Hello and welcome to the Genetics Podcast. I'm your host, Patrick Short. My background is in population genomics and studying the genetic causes of rare disease. I did my PhD at the Sanger Institute in the University of Cambridge and have been in biotech since 2018 when I started Sonogenetics. Sonogenetics helps academic and industry researchers to run large-scale genetic testing programs that speed up their clinical trials, generate data sets for the next big breakthrough, and give participants the best possible experience taking part in research. Each episode of the Genetics Podcast, we bring you insights from the leading minds in genetics and precision medicine, including household names and Nobel Prize winners, as well as early career scientists and biotechs working on the next big breakthrough. Whether you are a scientist, entrepreneur, executive, patient advocate, or simply someone curious about how genetics shapes our world, you're in the right place. Thank you for listening, and let's get started. Hello, everyone, and welcome to the Genetics Podcast. I'm very excited to be here today with Nadav, who is the director of the Institute for Human Genetics at the University of California, San Francisco, and a professor in the Department of Bioengineering and Therapeutic Sciences. When I was a PhD student, I actually read every single paper that came out of his group because I was interested in the non-coding genome. It was my focus. And at the time and still today, Nadav's group was churning out some of the most fascinating, creative, and thought-provoking science in this space. So I'm really excited to have this conversation today after knowing you for a number of years.
Thanks, Patrick. Great to see you again and looking forward to it.
I'd love to start. Your lab at UCSF focuses on gene regulatory elements, as I mentioned, and in particular, you look at their role not just in human disease, but also diversity and evolution. And I'd like to just start with a deliberately broad question, which is you've been focused on the regulatory genome, non-coding genome for a long time. How has our understanding of it evolved over your career? What do we know now that we maybe didn't know before? And then equally, what are the big open questions still that keep you up at night?
Great question. Yeah, when I started my sort of scientific career as a PhD student, sort of the genome was just coming out at the time, and I was fascinated that we don't know 98% of it, which is sort of the non-coding genome, which was even called junk DNA at the time. And so at that time, I chose a lab to do my postdoc that specialized in the non-coding genome. And all we had were three genomes at the time. It was human, and mouse, and fugu fish. So what they did was basically comparative genomics to find regions that are conserved in the non-coding genome and that could be functional and could be regulatory elements. So we would find these and then we would test them one by one for functionality in mouse enhancer assays, which take a long time and a lot of effort. And so, and that was sort of the only way to find them was this, three genomes and evolutionary comparison. So since then, it's really been an explosion. One, we have many, many more genomes recently with the Zoonomia project also, right? Publishing hundreds of mammalian genomes. But two, technologies that can find gene regulatory elements in a really genome-wide manner, like ChIP-seq, ATAC-seq, and all of these other technologies. The last missing piece that it was sort of an honor to be helping with and finding is really functionally characterizing them. So all of these technologies are descriptive. If you find a regulatory element based on a ChIP-seq peak or a ATAC-seq peak, they say that this could be a regulatory element, but you really need to functionally characterize them. And sort of the missing piece there was massively parallel reporter assays, where we could test hundreds of thousands of sequences in one experiment for the regulatory activity. And so with that in hand now, I think we could do a much better job on characterizing these and trying to see how variation in them leads to phenotypes. And now with CRISPR, also in high throughput screens with CRISPR, we can even test them in the genome. So it's beautiful to see how the field has grown. I still think there's a lot of room to grow. We're doing a much better job now in predicting regulatory elements. We're improving in predicting the effect of variation in them, but that still needs to improve and the code of them. And I think there's much, much more exciting stuff to learn over the years now with all of these technologies.
Yeah, absolutely. We understand the coding grammar pretty well. The triplet code is really well understood, but I think one of the open questions that you kind of alluded to there is what is the the grammar or the language by which regulatory elements operate. And as far as I think everybody is aware, it's far different and probably more subtle and more difficult to characterize. I wonder if you could talk a little bit about what we've learned about what the, how do you go actually from sequence to function in a regulatory element? And what have we learned about what matters about sequence, what matters about position, and just our understanding of the non-coding grammar more generally?
Yeah, so on that front, we and others have taken a variety of different approaches to understand regulatory grammar. For us in particular, I like sort of the language approach, where one is if you don't know a language, but you know a few words, for example, you can start playing with them and seeing what happens. And so we've done that by taking basically known regulatory elements that we know work, like promoters, enhancers, and doing what's called saturation mutagenesis. So mutating every possible base, if you have an A in a certain position, changing it to a C, to a T, or a G, and then testing, hundreds of thousands of mutations in them to see how that affects their function. And from that, you sort of can learn their grammar, what affects their function, what makes them stronger, what makes them weaker, and so forth. And then we've also taken the opposite approach of building a language, where we take sequences that are sort of neutral, that we know are not regulatory elements, and start building regulatory elements on top of them by putting transcription factor binding sites in various ways, various orders, orientation, or even in terms of the strand specificity, and also DNA shape. And so we've learned a lot from that in terms of that, there's really, really, I would say, fluent in terms of regulatory elements. You can really play around and mess with the transcription factor binding sites for most of them. You can move them around. They do have an effect in terms of order, but it's dependent on the type of transcription factor and the location sometimes of how close they are to a promoter. And so with that, I think we've learned a bit more about the code and sort of how they function, but there's much more to learn, I think, also within actual endogenous locus, where they also have a lot of various histone marks and various other marks that might affect them.
One of the kind of common paradigms that I think you hear, which I'm sure is, maybe roughly right, but probably precisely wrong in some cases, is the reason why we see so much of the non-coding or regulatory genome in common diseases is because most of the changes to the regulatory genome are relatively subtle in their impact on, protein quantity or expression timing or whatever it may be. And so that lends itself to the kind of common variant, common disease paradigm where lots of small changes can stack up, and it's hard to pinpoint any one, but they kind of ripple through the regulatory genome. And then on the flip side, in Mendelian disease, we see a lot more common disease variants because the effect sizes are a lot bigger. But there are notable exceptions in the Mendelian space, and also obviously in common disease. Does that paradigm feel roughly right still, or is it missing something? And if so, what is that something?
Yeah, I think for non-coding, we still don't have a good grasp of rare variation in that effect. And I mean, you've done beautiful work on that with neural disease, looking at 8,000 families, right? And you do see an enrichment for rare variants, de novo rare variants, right? And ultra conserved elements or sequences that are more highly conserved in the genome. And I think we need to do much more on the rare variant side. I think they'll have likely more of an effect in our assays also, in these massively parallel reporter assays, versus ones that are common. For example, we recently did, and this is a collaboration with Katie Polar's lab at Gladstone UCSF, testing a lot of GWAS and EQTLs and other potential variants that are common variants associated with psychiatric disorders. And we tested over 17,000 variants. We only saw an effect for 168, so a really low number from that in our assay. And so, one, it's a single nucleotide change, but two, I think it's common variants are more subtle in their effect versus rare variants in regulatory elements. And so now what we're doing, for example, is testing almost 100,000 variants from autism spectrum disorder de novo mutations. And so I think there I predict we'll see much more of an effect from the rare variants. Still not as high, of course, as exonic variants or so forth, but definitely believe we'll see a bit more of an effect for rare variants. And collaboration with Ryan Hernandez also at UCSF, we're doing a similar thing to show that I think rare variants would have a bit more of an effect in these. with all of that, yeah, I mean, I think the effects in regulatory elements are more subtle, but there are cases like the sonic hedgehog limb enhancer, the classic one, where you see a lot of sort of point mutations that do lead to limb malformations in humans, pancreatic agenesis, where there's quite a lot of mutations that lead to that in a specific enhancer. And so I think over time we will find, and I think that's another issue in our field, is the differences between these, why some enhancers lead to more subtle phenotypes while others, where you have mutations, lead to much more observable phenotypes. And it could be different types of enhancers or different ways they regulate.
Yeah, you mentioned ultra conserved elements, which go back to the days that you were talking about in the very beginning when you just had a couple of genomes and could look at some of these parts of the genome that were almost identical or completely identical, going back hundreds of millions of years. I think you've also done some work in human accelerated regions, which are kind of special case of these ultra conserved elements. I think these are not on the radar of most people who don't live in this, non-coding regulatory world, but I just think they're so fascinating. I wonder if you could talk a little bit about what those two concepts are, and then a little bit about what what your research has found there.
Sure. So ultra-conserved elements were characterized by Gil Bejarano's group and David when he was in David Hausler, and he was a postdoc at the time. And these are sequences that he and David termed as sequences that are conserved between humans, mouse, and rat for at least 200 base pairs, that there's no change at all in the nucleotide level when you do the comparative genomics. And they characterize about 700 elements that are ultra conserved, and many people have been trying to understand why they're so conserved. In my postdoc, we've actually knocked out 4 of these elements and surprisingly did not see an effect, including one that was 700 base pairs long. Later on, my sort of postdoc lab continued that work and characterized them and did see some subtle effects in some of these, but really not, as you would expect, like from such a conserved sequence to lead to that. Human accelerated regions were characterized initially by Katie Pollard and David Hausler's lab, and also Jim Noonan, when he was in Eddie Rubin's lab, characterized these similar sequences that are conserved in evolution. So, for example, conserved in all mammals, but change significantly in humans, and only in humans you see much more significant changes than by chance and over evolutionary time. And those are intriguing in the sense that they may lead to, why we are human. And a lot of them are actually near psychiatric disorder associated genes, which suggests, it helped us in terms of our brain involvement, but also, could lead to various diseases that are unique to humans. And so with Katie, over the years, we've been characterizing these sequences in a variety of different ways, both in mice or using these massively parallel reporter assays to characterize thousands of them, and also just the variation in them. If you change one nucleotide versus 2 versus 3, what does that lead to the change in the human accelerated region?
In these regions, but also in the regulatory genome more broadly, coming back to the grammar point that we were talking about earlier, I was curious to get your take on whether there is likely to be some kind of elegant organizing principle like the triplet code that somebody figures out, or if it's going to be messy and we need large language models maybe, or something non-deterministic to figure out what impacts have. My instinct is more the latter, but it would be amazing if it were the former. And I'm just curious on your take on it.
Yeah, no, I think it's going to be the latter also. I think it's much more complex. And we see, there's been a lot of various models proposed on function of regulatory elements that have been sort of shattered in various ways throughout the years. So it's definitely going to be more complex, and there's a lot of different rules that keep coming up and changing and so forth. I'm really, though, excited about machine learning and the ability of machine learning to improve. And we've done a lot of work recently on that, using a lot of our massively parallel reporter assay results to really try to improve both calling a regulatory element and the effect, variant effects. And I think the more data is out there, the more these tools can learn. And so I think that's where I'm hopeful that will improve in terms of understanding sort of the code and the effect of variants on it.
How much do you feel like the field is held back by the fact that to understand regulatory elements, you really need ideally every tissue at every time, because we know these things are kind of often exquisitely controlled, where they're only on in the developing brain and then they shut off, and they're only on in the heart and then they shut off. And some of these tissues are extremely hard to get access to in any large scale. But there have been big efforts to do this. So I'm wondering, like, but the matrix of tissues and time points is very large to fill out. How much of that is also, potentially understandable through machine learning, or is it something that we need to have some concerted effort to do a huge amount of primary tissue collection to figure it out?
Yeah, so on that front, I think, I mean, there's been great efforts now with genomics and single cell to characterize a lot of, with single cell ATAC-seq, for example, a lot of cell type specific regulatory elements by many, many groups, which is great. And the data is great to have. And definitely machine learning can fill the gaps for a lot of these in terms of trying to find ones in tissues that, are hard to access or so forth. Another gap that I think we need to fill is some regulatory elements respond to various inputs. So, for example, you
Right.
give a drug and then you have a regulatory element that comes up, or you're infected by a virus and that leads. And so there, I think that's going to be a harder one, in particular to characterize in very specific tissues or cell types. But I think that's another gap that we really need to start filling out for many various diseases also, which will be an important one.
Yeah, it's really interesting. It made me think of there's some news recently about Lilly making a pretty big investment into a company focused on long non-coding RNAs called Haya, H-A-Y-A. I don't know if they're on your radar, but my understanding of it was that they were looking for long non-coding RNAs that were unique to really specific cellular states, whether they be
Mm.
indicative of a disease state or potentially protective. And I thought that was a kind of interesting application. They also did the really smart thing of branding it as the dark genome rather than
Mm.
junk DNA or
Yeah.
non-coded genome, which I thought was pretty good.
Yeah, definitely. And I think also you can imagine using this for even, cell-free DNA diagnostics, right? Looking for long non-coding RNAs or short non-coding RNAs that are indicative of cancer or Alzheimer
Yeah.
or so forth that we could find.
You on the subject of biotechs, you published a really amazing paper years back that I remember reading that was focused on non-coding elements in Mendelian disease, but in a way that is maybe indirect, not about treating patients with non-coding causes of Mendelian disease, but actually ways to treat a very large class of often kind of devastating pediatric diseases as a result of haploinsufficiency, which is where you lose a copy of a gene for which you need really two copies to have full function. And you all came up with a really clever way of using CRISPR and non-coding elements to potentially treat this class of disorders, and you spun a company out as a result. I'd love for you to talk a little bit about that research and then a little bit more about where you're at with that today.
Sure, yeah. So we basically thought about haploinsufficiency, and I've been thinking about that actually years, even before CRISPR came out, of how we could basically try to potentially cure these diseases. These diseases basically is where you have one copy of a gene that's fine, another copy that's sort of non-functional. And so you now only have one functional copy of a gene that's giving you 50% of the RNA and 50% of the protein. And surprisingly, this is fine for many genes, but there's 660 genes that lead to human disease due to haploinsufficiency. And some of them are devastating, like cancer, neurodevelopmental diseases, polycystic kidney disease, and many others. And then if you look at the whole genome data sets, also, it's an estimated like 3,230 genes that never have a heterozygous loss of function. So the number could be even bigger. Some of these, of course, could be lethal, and that's why we don't see them. So the idea that I had way back was we still have one existing copy. It's just giving us 50%. So if we could boost sort of that copy and squeeze out more RNA from it, we might be able to rescue these diseases because that copy is normal. It's functioning well. It's just we're sort of giving what it can for now. And so when CRISPR came out, we thought, initially actually thought of using zinc fingers before that, but then when CRISPR came out, it was just the perfect thing to go after this because it's easy. It's like a software where you could just change. And so we decided to go after this. And basically, I was lucky enough to have an amazing postdoc in the lab, Navneet Matharu, who's now the CSO of our company for this. And Navneet, basically, we decided to tackle something that's quantitative, something that we can measure, that even if we upregulate that existing gene a little bit, we might see an effect. And so we chose obesity as a phenotype. And Navneet went after the two top obesity genes in humans, MC4R and SIM1, which together account for almost 7% of the population of the top 1% of body mass index. So it's quite a lot of people. And so the mouse models are the same. If you have one copy of this gene, you have mouse models that are extremely obese. And Navneet beautifully showed that if you target the promoter of a gene or even an enhancer of the gene, we can upregulate the existing copy and completely rescue their body weight phenotype. And we've done this both in a transgenic manner and also in using adeno-associated virus injections into the brain, which are more therapeutically relevant. And so following that article, there's actually 7 companies that have been formed now to use that approach to try to rescue haploinsufficient diseases. And one of them is one that we formed called Regal Therapeutics, that Navneet is now the CSO of that company. And we have now over 35 people working both in Berkeley and in MIT on it. And they've been focused both on neural disease and muscular dystrophies to try to help using this approach. And for me, if I help one patient in the clinic, I'll be the happiest person in the world. So I'm looking forward to seeing the results, not only from our company, but all the other companies, in the hope that will be helpful for patients.
Yeah, amazing. What was that like, starting the company and spinning it out? Was that the first time you'd done that, or had you spun companies out before?
Yeah, so that was the first time. So it was a lot of a big learning curve, as you probably know, also. And we're lucky at UCSF, we have a lot of support on that front. So we had this course called Startup 101 that me and Navneet took. That's a whole 4 months of intense and both marketing and figuring out the market research and having tons of people come and give talks to you, and working nonstop and forming a team along with basically students from Berkeley, from Haas School of Business, and from Stanford and others. So we put together a team, worked for 4 months, and then at the end of it, you present to a room full of VCs and angel investors and so forth. And the top 2 teams actually go forward with angel investing and so forth. And we were lucky enough to be sort of the second place in that. And went forward with that. We've given, I think, over 150 pitches over the years until we managed to get and secure funding. And we were lucky enough in the end to actually merge with another company, Regal, which were developing a lot of basically cell type specific regulatory elements that would drive therapies to very specific cell types. So now we have the combination of that, a cell type specific sort of driver, with sort of the CRISPR as a therapy, which is really nice, I think, in terms of clinical applications.
And the cell type specific driver, if we took the application in the brain, for example, is that because you may not want to actually boost activity in every brain cell, you need some kind of cell type specific? Maybe you could talk a little bit more about that.
Yeah, so the idea there is to drive, if it's CRISPR or if it's zinc fingers or whatever the therapy is, or just standard cDNA gene replacement therapies, to drive that gene, transgene, only in that tissue. And for that, we could use, all the enhancers and all the regulatory elements that we've been finding that we know are, for example, cell type specific, like only in excitatory neurons or only in microglia or so forth. And so we take advantage of that. Another nice thing that I didn't mention, but we found with our transgenic approach for the CRISPR-A. So when we rescued the obesity phenotype, and we also did it other than adeno-associated virus injections into the brain as a transgenic approach, where we had our CRISPR-A being ubiquitously expressed throughout the body with a strong CAG promoter. And so technically we could be upregulating and rescuing and upregulating this gene that we targeted, in this case is SIM1, was this obesity gene, everywhere in the body. And so we checked, are we doing that? And to our sort of surprise and relief, we saw that we're only upregulating it where the gene is active. So this gene is active in the kidney and the hypothalamus. And when we target its promoter, we only see upregulation in the kidney and the hypothalamus. And we also targeted a hypothalamus enhancer for this gene with CRISPR-A. And there we only saw upregulation in the hypothalamus, but not in the kidney. And so we think it's open chromatin, and that's based on a variety of different CRISPR screens that show, for example, that open chromatin you get much better upregulation. So I think when there's open chromatin, that Cas9 and the CRISPR activation machinery can bind. And so for the hypothalamus enhancer, for example, it's only open in the hypothalamus. It's only active
Right.
there. CRISPR can bind, and you get upregulation in the hypothalamus, but not in the kidney. And so the regulatory element that you target also with this CRISPR-A therapy also provides you a tissue specificity. So you sort of have a double layer of therapy, or sort of double layer of protection for specificity here, tissue specificity, where you're targeting a regulatory element that provides you that tissue specificity. Let's say it's a brain regulatory element that will only turn on the brain. And then if you have something that's driving your machinery also only in the brain, it sort of provides you that extra layer of support. And that's been a big issue with standard gene therapy, where you have these sort of CAG promoters or, ubiquitous promoters that you would get your cDNA lit up in every possible cell type.
Right. And the CRISPR-A is for people who haven't read the paper, we'll link it in the show notes, but it's not actually editing or cutting anything, right? Maybe it's important to clarify that. It's kind of a fusion between CRISPR and P300 or something like that. Maybe you could talk a little bit about what the CRISPR is doing.
Sure. So that was beautiful work from when Stanley Key, Luke Gilbert, and in Jonathan Weissman's lab at UCSF, and also from Charles Gerbash and Duke, where they basically took the Cas9 and they did two mutations. So the scissors part of the CRISPR cannot cut the DNA. So then what you could do is you could use it as a delivery truck to bring things to a specific zip code in the genome. And what you could bring is an activator, for example, that forces the gene to make more RNA. In this case, we've used that even before it was published. When we heard about it, we thought it would be a great approach. And we used it to drive things to the promoter or to an enhancer of a gene to upregulate, to basically force the gene to do more. And the CRISPR doesn't cut, which provides you an added advantage also, because it's sort of no DNA scars. You don't get any DNA cutting. The concern for off-target is only that it might bind somewhere else and upregulate another gene. And we felt that's sort of a less minor concern than causing DNA scars.
Yeah, for sure. It's amazing, fascinating. You've also since founded a second company focused in oncology. I wonder if you could talk a little bit about that, and it's based on a paper that you put on bioRxiv relatively recently that we'll also link out.
Yeah, so on that front, I was lucky enough to have a postdoc who now has his own lab in Austin, and that came from Berkeley, called Hai Nagoyan, who is very knowledgeable in using fat cells. And fat cells are actually amazing in the sense that we could do a lot with them that I think we're not doing enough. So we have two types of fat in our body. We have brown fat that's sort of near the neck, and it burns energy, has a lot of mitochondria. And as we age, it becomes less active, and that's why a lot of us retire in Florida or other warmer places. And then we have the white fat that's all over our bodies, sort of the bad fat that stores lipids, and basically, if we have more of it, we're obese and we can develop diabetes, cancer, and so forth. But what's beautiful about fat that we thought we could take advantage are two things. One, you could take out white fat and make it more brownish, what's called beige fat, by upregulating a few different genes. We focused on one called UCP1. And now you're basically making a white fat into more of a brownish fat that's burning more energy and to heat and so forth. Two, everything's there in the clinic to play with that. So you have liposuction, where you can easily take fat from a patient. And a lot of plastic surgeries utilize fat actually to implant. So we thought we could take advantage in the middle. You could take out fat by liposuction, play with it, and then put it back. And that could become a great therapy for a lot of metabolic programs. And so we initially focused on obesity. We did quite a lot of work on there, trying to show that on, for example, you put mice on a high-fat diet and you transplant fat that we take out, make brownish or beige to start burning energy and putting it back. We can protect mice from a high-fat diet, and we've done it also on various other obese models like leptin receptor knockouts and so forth. But the one that we focus the most on is actually using it as therapy for cancer. And the idea is that, cancer, needs a lot of nutrients to grow, like a lot of glucose, a lot of fatty acids, and so forth. And so if we could engineer basically fat cells that will outcompete the cancer, we might be able to sort of starve the cancer. And it worked beautifully. We were surprised initially, like when we did our first experiments in cell culture, we thought we made a mistake with the media or something. We had to repeat it a few different times because we had so many cells die. But when we repeated it, we believed that it's actually real.
Huh.
And so we've done this now quite a lot in a variety of different cancer models, both xenografts and genetic mouse models, like a pancreatic mouse model that develops pancreatic cancer due to mutations. And we implant the fat next to the pancreas, and we see shrinking of the tumor there. We've done it for breast cancer, where we implant it either next to the breast cancer, next to the breast, or actually even in the back, where it outcompetes in both places. So it doesn't really need to be close to the tumor to shrink it. And then we're lucky enough to work with clinicians here at UCSF. So we're lucky enough to work with Jennifer Rosenbluth, who has basically over 2,000 mastectomies. And so in the breast, there's quite a lot of fat. So we could take from the same patient the tumor, which Jennifer grows as organoids in a plate. And on top of it, we can grow fat from that same patient, which we modify to become brown and show that we can shrink it. Another key thing for this is you're always worried when you're doing cell therapy or you're inserting something. And so we've also shown that we could control it with tetracycline, or working with Tejal Desai here at UCSF, who's a bioengineer who builds cell scaffolds. We build cell scaffolds, which are like a cassette where we grow these fat cells, put them in, and then can take them out. And that also led to suppression. And finally, cancer is smart. It will sort of outcompete initially, but then it will find another way, right? And it will find another metabolic program. And so we also show now that we can make these fat cells outcompete to various other metabolic programs. So for example, we took a pancreatic cancer that starts using uridine instead of glucose, and we make the fat cells outcompete for uridine, and we show that that also suppresses it. So I'm really excited about this approach. I think we're just in the beginning. We're now trying to build these cells even more, and we can upregulate many, many genes, not just one, to make them outcompete. They're great secretors, so you can imagine them secreting things against the cancer. And so I think we're just in the beginning of really utilizing this as a new cell therapy for cancer and many other metabolic diseases that we're starting to work on.
That's fascinating. And do you need to do it as a cell therapy where you kind of take it out and put it back in, or might there be a way to activate it locally? I'm thinking about, the cancer example. If you have a solid tumor or something, can you recruit the fat cells around it to do their thing, or how is that not going to work?
So yeah, it could be done. I mean, we've done it for the obesity, and another approach was just injecting AAV, adeno-associated virus, to upregulate UCP1, this gene, throughout the body. So it would upregulate all the fat cells and increase the activity of the brown fat cells also. And that also worked beautifully for obesity. The issue there is stopping it when you don't want to at the end. And so the cell therapy, I think, is more of a modular approach where I can put it in, take it out, and I can modify what it's basically targeting. But it's definitely doable, also just as a standard AAV injection. And we've shown that at least for the obesity work.
Yeah, that's amazing. Just as we wrap up here, what's on the horizon for you and your lab? Like how many people are you? Are you a big group, medium size, small, and what are the sorts of things you're focused on going forward?
Yeah, so we currently have about 25 people in our lab, and we've been focusing on, we've always tried to characterize regulatory elements. That's always been a theme. So in that regard, we're always trying to tie them to disease. We've always also tried to tie them to evolution, which we didn't talk about, but we work a lot on two main models for evolution. One is human evolution, which we talk a bit about, human accelerated regions. But we're also in collaboration with David Gockman in the Weizmann Institute, trying to characterize a lot of regulatory elements that are specific either to modern humans or to archaic humans.
Yes.
And to try to understand better how archaic lumens look like and various phenotypes in them. And we have big projects there. And then the second is bats. I've been fascinated with bats ever since I was a kid. And so we've been working a lot on bats to understand bat wing development, and lucky enough to collaborate with Nicola Ayling in University of Cape Town, South Africa. We can collect bat embryos that we could do genomics on, and also recently started working on bat diets, in particular on fruit bats, and trying to understand how fruit bats eat so much sugar and don't get diabetes, I've done a lot of genomics on fruit bats versus insect bats to understand that. So that's another major part of the lab, I'd say. And then the last is therapy, where we're growing not only in using gene upregulation to rescue the disease-causing gene, but upregulating genes that we think will have a beneficial effect
I was going to ask
for
about
the
that. Yeah.
disease. And for example, the obesity or sort of the fat for cancer is one example for that, but there are other examples where we're working on that. And then finally, I think for all of this, what we need also is better tools to control gene regulation. I would love to basically control a gene with my iPhone, basically, where I can dial the sort of the amount of the gene and turn it off when I can. And so we've been hard at work on trying to develop some things there where we could control things much better. And then you can imagine how that will be beneficial for therapy, for treatment, and even monitoring a patient or so forth in a variety of different ways. So that's also a big sort of push now that we're doing in the lab.
Amazing. I was actually going to ask about the upregulation point and also about the kind of brown or beige fat and linked to regulation, but you definitely answered both of them with one with one fell swoop. So what that's really interesting. I want to ask you a couple follow-ups about each of these because I think we've got a little bit of time and that connection is interesting. I've got to ask about bats. I think they're fascinating creatures as well. Are they are they particularly interesting from like a model system perspective, or tell us a little bit about that because I think they're super cool, but I don't know much about them as a model system.
So bats for me are like superheroes. I think each one has their own superpowers. So, anywhere from flying, of course, to echolocation, to blood sucking without coagulation, right, for vampire bats. And by the way, there's 1,400 species of bats. Only three are blood sucking. So they get a
Oh,
bad rap for being vampires. There's only three species. And the variety of bats is amazing. I mean, we go now, we're lucky enough to go every year to Belize with a trip that's organized by the Natural History Museum with 50 real bat researchers, unlike me, that do field work and capture bats. And we go there. It's an amazing place. It's in Lamanai, which is Mayan ruins. And so a lot of people have been digging for gold there. So there's many caves for bats.
wow.
And we basically catch bats around these amazing pyramids. And because of that, there's such a variety of bats. Like this year, we caught over 1,123 bats, 38 species. And just to see the variety and the differences between them is just amazing. And so they're mammals, of course. And so I think there's a lot that we can learn from them. And on our side, we've been focusing on diets because they're so extreme. And in particular, the fruit bats lower sugar levels like nothing we've seen. So if you compare, for example, humans or mice, where it takes time to lower glucose levels, these guys, they go up and go down right away. And so what we're trying to figure out is what they developed to do that. And so, for example, on that front, we've done a lot of single cell on pancreas between fruit bats and insect bats. And we find, for example, they have much more alpha cells and beta cells that control glucagon and insulin. But we also find, for example, if we look, and we've done combined single cell, so we can look at the ataxic peaks, and you look at the insulin region, for example, you see beautiful, very nice high peaks versus the insect bats, suggesting that they figured out ways to really have this sort of poised and ready to go
Mhm.
and control the insulin and the glucose really nicely. And so we've been basically trying to understand how they do that also on the regulatory element level. And you can envision, for example, using those regulatory elements later in the future for therapy, in a sense, to
Yes.
control insulin much better or so forth. So that's what we've been focusing on in that regard.
That's fascinating. That is I all of these tree of life programs that are looking at sequencing, some people don't find them that interesting. I find them to be one of the most fascinating things. I think we're going to learn so much from sequencing the tree of life.
Mhm.
I don't know what it is going to be yet, but I'm just confident that things like
Mhm.
this we're going to learn so much from these programs. We probably already have already from the ones I've seen.
Yeah, and I mean, even if you think about Ozempic, for example, right, GLP antagonists, that all came from the Gila monster in the sense that basically manages to paralyze the prey by having a 3-hour long half-life of GLP, right? So that was the first time that people thought that this could be a therapy. And that was actually the first therapy coming from a Gila monster. The sequence of the GLP antagonist was that. So beautiful example of how we could use sort of the tree of life to learn about a variety of different things and develop therapies for a variety of different diseases.
I did not know that. I also want to ask about the evolutionary point. I listened to a really interesting podcast recently with David Reich talking about a lot of the common understanding of early human evolution being overturned and or at least modified by all of our understanding from ancient DNA. What's the high level of the work you're doing in that space, and what are you learning right now?
Yeah, so the majority of differences in hominids between archaic humans, Neanderthals, Denisovans, and modern humans is non-coding. Of course, most of the variation is in the non-coding space, and we really don't know what it does. And so, in the past, in collaboration with David and also Dmitry Petrov and Hunter Fraser, when David was a postdoc there, we characterized all the modern human variants, and because they're non-coding, we just did these massively parallel reporter assays, testing thousands of them in different cell types. So in bone cell types or in stem cells or in neurons, and trying to see, one, are there regulatory elements? And two, does the modern human lead to a difference versus the archaic? And we learned a lot and found a lot of cool regions that we think are important both for human evolution and also human disease. Now we're sort of doing the opposite. We're really interested in the archaic, since we don't have anything but fossil records for them. And so we're basically taking all the archaic specific variants that we see only in the archaic humans and don't see in modern humans or in primates, and then testing all of them in these cell types. And now we're starting to learn of a lot of cool and interesting traits that these archaic humans may have had, which we could not learn, right, based on fossil records or genomes. So
like what sorts of things?
So for bone, for example, we're starting to find regions where we test the archaic sort of sequence and see differences in its activity in bone. Then we look, of course, what gene does that regulate? And then we could look at differences in bone condensation, for example, between the archaic versus the modern human. And what's nice there, that's known based on the fossil records. And we're just literally now analyzing the neuronal results. So hopefully stay tuned, but it'll be really interesting to see what we get there. And for the neurons.
This is amazing. The breadth of work you're doing is incredible. How do you decide what to focus on and what not to? Obviously, you have this common theme of regulatory elements, which is a helpful organizing principle. But you're covering such a huge amount of ground. It's amazing.
Yeah, I mean, I wouldn't be able to do one thing forever, and that's maybe a good thing or a bad thing. But yeah, for us, it's always been gene regulation, and really depends. It's important for me that people be motivated and excited about what they do. So it really depends on the person who joins the lab and what they want to do, and then we take it from there and grow from there.
Yeah, it's amazing. This is you get the award officially for the episode that has covered the greatest breadth of ground. I'm honestly amazed by this. The previous record holder, he doesn't know, but was George Church on one of the previous episodes, who also has a famous breadth of interest. But we have covered so much ground. I could talk to you for hours. We don't have that today, but I just want to say thank you for sharing all this fascinating science with everyone. And it was really nice to reconnect after a couple of years since I've seen you at ASHG.
Thanks. Great. And great seeing you again.
Great. Thanks, everyone, and we'll see you next time.
That wraps up this week's episode of the Genetics Podcast. I'd like to give a huge thank you to our guests for sharing their valuable insights and experience. And thank you as well to our listeners, as always, for tuning in. If you enjoyed today's talk, the number one thing I would really appreciate from you is if you could share it with a friend or colleague who you think would enjoy it as well. We would also really appreciate if you could subscribe to our show and give us a quick rate and review on Apple Podcasts, Spotify, or your favorite podcast platform. Both of these things help us become more visible when people search for genetics and precision medicine podcasts. And we're always eager to hear from you. Please reach out to us with any questions or feedback on social media. You can find Cynogenetics on Twitter, LinkedIn, or visit cynogenetics.com. Finally, a big thank you to the team behind the scenes who make all this possible. In particular, Amy Cousins and Sonia Shah, who produced the show, and James Pierce from Selected Frequencies, who handles the audio engineering. I'm Patrick Short, your host, and it's been a pleasure. Thank you, and we'll see you next time on the Genetics Podcast.